Hello, everyone. Um, Today, let's start on the chapter 3, okay? The bacteria and the archaea. Where in this section, we are going to study the structure of the bacteria and bacteria, okay? Uh, when we study, when we study the chapter 1, 2, so we have studied three groups of microorganisms, right? So those are the bacteria, archaea, and the eukarya, right? Now, so what is the difference between the bacteria and the eukarya? Well, the difference is that eukaryotes, eukaryotic cells have the real nucleus and and complicate and complex chromosome. Also, the cell wall is different. Now, also the eukarya has real organelles. So now let's talk about this one by one. So the structure of bacteria. All the bacteria have cytoplasmic membrane. Okay, and cytoplasm. Cytoplasm is a gel-like liquids inside of the cell. Okay, and all the chemical reactions happened in the cytoplasm. Bacteria also have ribosomes. Ribosome is organized for the bacteria to produce proteins. Cytoskeleton. Cytoskeleton are made of actin and microtubule. Okay, actin probably you guys know that from anatomy and physiology courses. So we have actins in our muscle tissues. Okay, so the actin is one part of the proteins that so that allows our muscles to contract and give our the locomotion of our limbs. Okay. Yeah, so the cytoskeleton of the bacteria gave the shape of the bacteria. Okay, gave the shape of bacteria. One or few chromosomes. Yeah, so the chromosomes can contain the DNA. So DNA restores the so the DNA restores the DNA stores the hereditary information for the bacteria. So most bacteria have cell wall, but not all the bacteria. Okay, a surface coating called glycocalyx. Some bacteria have flagella, pili, and fibria. Okay, flagella is specific bacterial organs to provide the locomotion for the bacteria. An outer membrane. So the outer membrane is specific for the gram-negative cells. Okay. Nanowire or nanotubes. So we talk about this later. Plasmids. Plasmids is a small circle DNA. So bacteria use the plasmid to transfer the DNA information between the cells, between different bacteria, inclusion and endospores. Okay, microcompartments. Now let's take a look at this diagram. Okay, so this diagram shows different part of the bacteria. As we can see, so this is the flagella. So this thin green tubes. Okay, salt layer. So this is old membrane. Okay, so So the yellow one is the cellular membrane. So the dense red one is cell wall. Okay. So we talk about this. So we discuss this later on. So most bacteria actually they live independently. Okay, so they don't live together. However, some bacteria they live together. Okay, they act as a group in colony and bear films. So the bacteria can also communicate each other through the nanotubes. Okay, so they can transfer the signals through the nanotubes. They can also transfer the plasmid. That means they can transfer the gadgetry information through the nanotubes. So the bacteria is very small. Okay, so its size is about 1 micron. Okay, um, cocci is about 1 micron. Robs, so the length is 2 micron. The wide is 1 micron. Okay, yeah. So anyway, it's about 1 micron. Okay. Pleomorphism. That means different bacteria have different have all kinds of size and shapes. Okay, so based on the size and shape, so we can classify the bacteria to different categories. Since why the bacteria have different size and shape? Because this is determined by the by the inheritary information in the DNA. Okay. So so here is pleomorphism. Okay, so this is the coccus. Okay, coccus. So this is the coccus. This is a robe. This is the robe cells. So this is the barbary. Okay, this is a spiral. So this is spiralium. So we talk about this one by one. Now let's about cocci. Okay, so the cocci is single. Okay, so the cocci doesn't. So the cocci normally so they live alone. Okay, if two cocci live together, attached to together, so it is called diplococci. Okay, that means two cells pair together. Tetrad. That means groups of four. Okay. Staphylococci or the micrococci. So those are the irregular cluster. Okay, many cells stay together. Streptococci. So there's a chain. So several cells link by the hand and tears to form a chain form. Okay. Sacina is a cubical packet of 8, 16 or more cells. So as we can see in this diagram, so if the cocci divided in one plane, so this is the diplococcus. Okay, that means two cells. Okay, so if many cells link together by the hand and tears to form a chain, to form a chain, so this is streptococcus. Okay, streptococcus. However, if the bacteria divide into planes, so they can form tetrad. Okay, tetrad. So that's a group of four cells. So if excuse me, so if they divided in two planes, okay, so tetrad is still one plane. Okay, so if they divided in two planes, so they can form Sacina. So Sacina is 3D structure. Okay, So it is formed with 8 or to 64 cells. Okay, so this is 3D structure. So we can see, so this is the succinct, okay, maybe 64 cells, okay. So if the cells divided in several planes, okay, so this is also the 3D structure, okay. So we can have a cluster of the cells. It's like a grip, okay, cluster of grip. So we call this irregular cluster, such as the staphylococci and the micrococci, okay. So now let's talk about the bacilli. Okay, bacilli. Bacilli is also single cells. Okay. However, two or more cells can stay together to form complicated structure. Okay. If two cells stay together, we call this as the diplobacilli. Okay. So this means two cells pair each other. So if the cells link together to form a chain, okay, so we call this as the streptobacilli, okay. Um, palisades, okay, so there's a series of chain, okay, touched by a small hinge region at the end, okay. Now let's talk about the spirali. Spirali. Spirali is occasionally found in a short chain, okay. Spider threads, so there's series so that twist their shape, okay. Now let's work on this question, okay. Which of following structure are possessed by some but not all prokaryotic cells, okay. You can see ribosome. So cells, so the bacteria have to have this one because they need this to produce protein. Okay, cell membrane. Okay, chromosome. Yes, so all the bacteria need all these three structures because uh, without the either one, so the bacteria cannot survive. Okay, so the flagella. Correct answer is the flagella. Okay, because some bacteria have this one for the locomotion. Okay. Describe the structure and the function. Okay, so now let's work on the section 2. Okay, so let's discuss the different structures. So the external structure provide the motility for the bacteria, also provide the function of attachment to the external so the external materials such as stone, glass, all the bare films. Okay, and now so as what we have talked about, so the flagella provide the motility. Okay, also the axial filament. Okay, so the fibrin and the pili and nanotubes provide the attachment. Okay, so the flagellum is primary. So its primary function is for the motility. Okay, so it has three parts. Okay, filament, hook, and basal body. As we can see this diagram, so this is the filament. Okay, so this filament is a very long thin tube. Okay, so this is the hook. Okay, so it's like the hinge. It's like hinge, and to connect the filament and the basal body. Okay, the basal body. So the basal body is the part in the cell wall and the cell membrane. Okay, so it can provide, so it can twist, so provide the um, so provide the power to rotate the filament. Okay, so this is the electron microscope view of the filament, so of the flagellum. Okay, the basal body. Flagellum. So the flagella is the polar arrangement. That means flagella attach at one uh, at one or both ends of the stairs. Okay. So based on the different attachment to the stairs, so we can divide the stairs to monotrichos. Monotrichos means single flagellum. Okay. Lophotrichos. That's small bunches of flagellum emerged from the same size. Okay. Amphitrichos, the flagellum. So the flagellum attached to both sides of the bacteria. Okay. Peritrichos. So the flagellum are dispersed randomly. Okay, randomly over the surface of the stairs. So so this one is the monotrichos. Okay, this one is the monotrichos. Monotrichos. Okay. So this is maybe so this is the Lophotrichos. Okay, Lophotrichos. Okay. So now let's talk about the how does the flagellum provide the locomotion for the stairs. Okay. Actually, so the stairs, because the bacteria can move to certain directions. Okay, how does the bacteria find their direction? Because they use chemotaxis to find their direction. Actually, the chemotaxis means the bacteria respond to the chemical signals to determine their directions. Normally, so this nutrition's gradients provide the chemicals signals because the bacteria have to find their foods. Okay, so they can move to the high concentration nutrients area. Okay, so so actually there is two chemotaxis. One is the positive chemotaxis. So that means normally normally uh, nutrients so that's nutrients gradients. Okay, so the bacteria will move from low concentration nutrients gradient area to the high concentration nutrients gradient area. Negative chemotaxis, so that means move away from high concentration gradient area, such as the toxins for the bacteria. So if the bacteria detect some toxins because those all the antibiotics, so because those chemicals can kill the bacteria, so the bacteria once the bacteria detected those substances, so they will move away from those toxins or antibiotics. So how bacteria move? So they can run their so they can run their flagellum. Okay, so they can rotate the flagellum clockwise. Okay, so that will cause a smooth linear movement, a smooth linear direction movement. Okay, stumble. Okay, reversal of direction. Tumble. Okay, reversal of the direction of the flagellum. For the run, okay, so once the bacteria detect substance concentration gradients, so they can use these gradients as the direction, so they can move as a small linear direction. Okay. However, if the bacteria doesn't recognize or found um, a strong chemical substance gradients, so the bacteria will move tumbly. Okay, that means the bacteria can move this like this to different direction to to search the to search a substrate. Okay, once they find the substrate concentration gradient, so they can move, so they can run, so they can run linearly to the high concentration of to the area of high concentration of substrates. So this is the linear direction running. Okay, straight. Okay, so this is tumble. Okay, tumble. So the bacteria can twist their directions. Okay, so they can walk around to search or run away from the substance. So this is still the tumble. Okay, tumble. This is run. So tumble we can see. Tumble, so we can see the flagellum. So the flagellum of the bacteria can 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 move individually. Okay, can move individually. So that causes the bacteria move irregularly. Okay, irregularly. However, for run, okay, so all the flagellum actually move together at one direction. Okay, so that causes the bacteria to run to one certain directions. Okay. Actually, so all these all the template or run movement actually help the bacteria to find the substance and run or move away from or run to or move away from the substance. The spiral thread is the corkscrew shaped bacteria. Okay, so the bacteria twist their body. Okay, so it's so possess unusual and regularly mode of locomotion. So the periplasmic flagella, okay, periplasmic flagella, so also called axial filament. So this is the internal flagellum enclosed in the space between the cell wall and the cytoplasmic membrane. So the fimbria, okay, fimbria is a small bristle-like fibers, okay, sprout of the surface of the bacterial cells. So bacteria can use the fibrine to attach to the external materials such as the glass, the plastics, or the rocks. Okay, so bacteria can use this structure to localize themselves to the external materials. Okay, and to avoid a washout by the water streams like that. Okay, so allow tight additions between the fibrillum and the epithelium. Okay, bacteria colonize and so bacteria can also use. So once the bacteria is tightly attached to the materials, so they can continue to grow and infect the host tissues. Okay, for example, so the sugar buds, okay, sugar buds. So the bacteria in our mouth can use the fibrillum to tightly attach to our teeth. So once the bacteria tightly attached to our teeth, so they can secrete the acid. Okay, so they can digest, eat the sugars in our mouth and secrete the acid to destroy our teeth. Okay, the pillars, so the plural is pillar. Okay, it's also used in the conjugation between the so it's used to in the conjugation between the bacterial cells, especially in the gram-negative bacteria. Okay, so type 4 pillars can transfer genetic materials, act as fibrillum, and assist in attachment. Okay, so they can also they can also let the bacteria attach to the external materials. Okay, some can work as the flagella and let the bacteria to move. Okay. Nanotubes, also called nanovirus. Okay, so that's a thin, long, and hollow tube. Okay, extension of the cytoplasmic membrane. So bacteria can use the nanotubes to link their cytoplasm together. Okay, so they can use these nanotubes to exchange some materials in their cytoplasm. Okay, so because they nanotubes work as the channels, okay, to transfer such as amino acids or the harvest energy, okay, by shutting. Or also they can communicate with by using electron waves. Okay. This is called breathing rocks instead of oxygen. Now this is the electron microscope view of the conjugation process. This is a pili. Okay, pili. So as because we know that bacteria can use pili to exchange some materials. Okay, exchange transfer genetic material. Okay, pili. So now let's talk about the S layer. Okay, S layer. S layer is the single layer of thousands of copy of single proteins linked together like the chain layer. Okay. So this is only produced when bacteria in the hostile environments. Okay. glycocalyx. Okay, glycocalyx is the coating of repeating polysaccharides glycoprotein unit. Okay, um, polysaccharides means so this is a chain of a sugar, all the sugar protein. Okay, glyco means the sugar, so that means this is the protein with the sugar chain. Okay, so both can be used as the so polysaccharide and glycoprotein can be used to receive the secondary signaling. Okay, signaling pathway. Okay, slime layer is so slime is the sticky liquid. Okay, so this is loose materials. Okay, so their function is to protect against loss of water. Okay, so it's like so it's like a sticky candy. Okay, so it can so it can um, uh, so you can store the water inside of cells to prevent loss of the water and nutrients. Okay, so the capsule is the tightly bound dense sticker structures produce the sticky so can stick so that so it can stick to the material such as the agar. Okay, to help the bacteria to form the colonies. Okay, so as we can see, so this is glycocalyx. Okay, so this is asli. Okay, so all these are actually um, other structures protect the bacteria to to prevent the water or nutrients loss. Also provide have the functions to facilitate the bacteria to attach to the materials and to form the colonies. Okay. So the glycocalyx forms the capsule. Okay, and however, some bacteria doesn't have those structures, so those are called uncapsulated bacteria. Okay, so the capsule formed by many. So if the bacteria have the capsules, so actually many of these bacteria can cause disease to animals or humans. Okay, so they are pathogenic bacteria. Okay, so actually, so the capsule can cause diseases. Okay, so they have the pathogenicity. Okay. Because so the capsules, once the bacteria have the capsules, so they can so once this bacteria invade our human. Okay, because once the bacteria invade our human, so our neutrophils, white blood cells or T cells will capture those bacteria and eat them. Okay, so the capsules can prevent the bacteria to be eaten by the neutrophil or T cells. Okay, so they can protect against phagocytosis because the eaten by the neutrophil is called phagocytosis. Also, the bacteria can also generate some sticky materials, so it is called biofilms. Okay, biofilms. For example, So the cigarbugs and other bacteria in our mouth can generate the beer films. So the beer films can form the plaque on the teeth. Okay, so the plaque is very sticky. So once the beer film is calcified, so it's very rigid. Okay, like a stone. Okay. So the bacteria can, once the bacteria generates the beer film, actually, it's more like the bacteria make their own house. Okay, so the bacteria can stay in there. Okay, and to protect them, prevent washout or brush it out by our teeth brushing. Okay. So the beer film's function is responsible for the persistent colonize because it's very sticky, so it can let the bacteria attach to the material. Okay, plastic, metals, and other devices. Okay, it's like this diagram. Once the bacteria attach to external material surface. Okay, so they can generate the green stuff is the beer film, so they can generate the beer film, so they can generate more and more. Okay, so they can make more and more beer film. Okay, so it's like a house. So that looks like the bacteria mirror make their own house, so they can stay in there. Okay. So the other bacteria can also get in the beer film. Okay, so all the bacteria can grow in the beer film. So they can use beer film to attach to the such as plastic, metal, glass. Okay, even some medical devices. So they can attach to the medical devices and cause trouble. So now let's work on this question. Which of the following bacterial appendage is not used for attachment? Okay. So I give you 5 seconds so you can review the previous slides and give me the correct answer. 1, 2, 3, 4, 5. Yes, so the correct answer is B. Okay, so the flagellum actually, bacteria use flagellum to provide the power for the locomotion. Okay. So now let's work on the section 3. Okay. Section 3 is about the envelope. Is about the envelope. Okay. So what is the cell envelope? Okay, so this is the outside layer of the cytoplasm. Okay, so this is made of the 2 or 3 basic layers that each perform distinct functions. Okay, but together act as a single protective unit. Okay, so the 2 or 3 layers, these are the cell wall. Okay, cell wall, cytoplasmic membrane, and outer membrane. Okay, so some bacteria have 2 layers, some bacteria have 3 layers. Okay, so we can use the different composition to classify the bacteria to 2 different categories. Okay, so as we can see, so this type of cells have bacteria have 2 layers. Okay, 2 layers. So this is 1, so this is 2. Okay, so the 2 is the cellular membrane. Actually, this bilayer of phospholipid. Okay, so this is 3 layers. Okay, 3 layers. So this is outer membrane. Okay, outer membrane 1. So this is the cell wall. Okay, so this bacteria have thin cell wall. Okay, so the 3 is the cellular membrane. Okay, so the cell wall can determine the shape of the bacterium. Okay, because this is a very rigid material. Okay, provides a strong structure support to keep the bacterium from bursting or collapse. Okay, due to the osmotic pressures. Okay, because normally, so the bacteria live in a hypotonic environment, such as in the fresh water. Okay, uh, since the osmotic pressure inside of the seal is very high. Okay, so that attract more fresh water get into the seal. Okay, so once more and more water get into, so the bacteria will get bigger and bigger and eventually rupture. Okay, however, if the seals have the seal wall, since the seal wall is a rigid material, so it can prevent the bacteria to rupture. Okay. So certain medication can target on the cell wall, okay, to kill the cells. For example, the penicillin, okay. So once the penicillin can destroy the cell wall, okay, destroy the cell wall. So once the bacteria doesn't have its cell wall, so the water can continually get into the cells, so eventually the cells ruptured. Once the cells ruptured, so bacteria die. Okay, so this is the mechanism how penicillin treats the infections. Okay, so why the cell wall is rigid? Because the cell wall contains peptidoglycan. Okay, peptidoglycan. So peptidol means the peptides. Okay, so that's a short chain of amino acids. Glycan means the sugar. Okay, so this is the sugar peptides. So peptidoglycan compounds composed of repeated fragments of long glycan sugar chain. Okay, cross-linked by short peptides. Okay, short peptides. As we can see in this diagram, okay, so the peptidoglycan provides strong but flexible support framework, okay. So the purple stuff for this bacterium is peptidoglycan, okay. So we can see, so this is sugar. Okay, this is the sugar. Okay, so we can see so the peptidoglycan is made of multiple chain of the peptides. Okay, peptides. So the peptides is conjugated by a sugar. Okay, by a sugar. So we can see so the chain is same. Okay, the chain is same. LD, 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 LD. Okay, so the chain is almost the same. Okay, so the peptide chains is linked together by the glycine. Okay, glycine. Okay, so the glycine. Okay, glycine is also one type of the amino acids. So the peptides linked together by the glycine bond. Okay, glycine chain. Okay, so the glycine bond. So that so the glycine is more like a bridge. Okay, like bridge to link all the peptide chain together. Okay, so this provides a very strong. So that's made the peptidoglycan very strong. Okay, very strong. It's very substantial. Okay, to resist to protect the bacteria and resist the lysis induced by the by the by the by the by the hypertonic hypertonic so the hypertonic solution. So so the cell wall, since the cell wall contains peptidoglycan, so okay, and so if the bacteria has two layers, okay, two layers, so the bacteria have a very thick peptidoglycan, okay, so we can stain the cell wall, stain the peptidoglycan, okay, with gram staining, okay, so some cells with the thick cell wall, so they can be stained as gram positive, okay, so this is called a gram positive cell wall. So gram positive cell wall normally, so the cell wall is very thick, homogeneous sheet of peptidoglycan. So since the cell wall is thick, it contains tens of peptidoglycan. Normally it's 20 to 80 nanometers in thickness, okay, so this is very, very thick, okay, so it contains the teichoic acid and lipoteichoic acid, okay, so it can since this cell wall is very thick, so it can host host many gram dyes, okay, so it will be stained as the gram positive. So the gram negative cell normally so these cells have three layers, okay, three layers, so the cell wall layers is very thin, okay, very thin, so it contains less peptidoglycan, okay, it's about only 1 to 3 nanometer in thickness, so it's very thin, okay, so it's 10 times 10 or 20 times thinner than the gram positive cell wall, okay. Since it's very thin, so it cannot host too much gram dye, so it can be digested so it can be stained as gram negative, okay. So it, however, compared to the gram positive cell wall, so it's more flexible, okay. So the protection is weak, okay, since it's very thin, okay, so it's very sensitive to lysis. So because if the cell loose or because the cell wall is not substantial, okay, so the bacteria is sensitive to the lysis. So this is the green staining, okay. Green staining contains 4 steps. So first we dump the crystal violet to stain the cells, okay, and then we use the grams iodine, okay, grams iodine. So grams iodine stabilize the crystal violet dye, okay, to form a large complex in the peptidoglycan, okay, peptidoglycan, okay, peptidoglycan. So this is gram positive cells, so they have thick cell wall. They have more peptidoglycan, so they can hold more dye, okay. So the gram negative cells have thinner cell wall, so can hold less dye compared to the gram positive cell, okay. Then we use the alcohol to wash the bacteria, to wash out the dye, okay. Since the gram positive cells have thicker cell wall, so they can because they hold more dyes, okay. Even we wash the dye, however, many dyes is still held by the peptidoglycan, okay. However, for the gram negative cells, so things the cell wall is very thin. Also, the alcohol can destroy the outer membrane. Okay, because the outer membrane is still the phospholipid. Okay, phospholipid can be dissolved by the alcohol, so the gram-negative will lose their outer membrane by alcohol. Okay, so if the bacteria lose their outer membrane, so the cell wall is directly exposed to the alcohol, so the alcohol can wash out nearly all the dye of the crystal violet. Okay. So the step 4. So we restain the cells with saffron. Okay, saffron is the red dye. Okay, so once we add this red dye, so this red dye will this red dye will stain the peptidoglycan. So the red react with the blue. Okay, so this so this is violet. This is violet. So they can form a blue color. Okay, so they can form a blue color. However, since the gram-negative cell lose all the crystal violet. Okay, so at this type, so the gram-negative cells is staining as red. It's stained as red because the saffron is red color. Okay, so that means for the gram-negative cells is blue. Okay, blue. So for the gram-negative cells is red. Okay. So and then we can use the microscope to differentiate the gram-positive cells and gram-negative cells. So now let's talk about the no typical cell wall. Okay, since this is no typical cell wall, so we cannot use gram staining. We can use acid-fast staining. Okay, so we can classify these cells as acid-fast bacteria, such as Mycobacterium tuberculosis and Nocardia. Okay, so they contains peptidoglycan and so they have the peptidoglycan. Okay, can stain as a protein. However, they have the cell wall. So the cell wall is all so their cell wall are also made of the lipids. Okay, right? The lipids is actually is wax. Okay, wax. So the wax can protect the cells, prevent the penetration of the medications. Also can prevent the loss of the water or nutrition from the bacteria. Okay, so that's why. So those bacteria are very strong. It's difficult to kill these bacteria. Okay. So the lipid is called macolic acid. Okay, macolic acid is very long chain fatty acid. Okay, fatty acid found in the cell wall of acid-fast bacteria, contribute to the pathogenicity of bacteria. Okay, for example, the tuberculosis. So the tuberculosis. So since this is fatty acid like the wax, so they can prevent the medications to penetrate into the bacteria to kill the bacteria. So it's difficult to kill the tuberculosis. Okay, so this material can also cause fever, cause the symptoms to the human. Okay. So make the bacteria highly resistant to the chemicals, dyes, such as the antibiotics or the grain staining. Okay, because the grain dye cannot. It's very difficult for the grain dye to penetrate the peptidoglycan. Okay. So no typical steer wall such as archaea. Okay. So some archaea have steer wall composed entirely of polysaccharide. Okay, so this is polysaccharide. So this is the sugar. Okay, this is so they have the sugar steer wall. Okay, sugar steer wall. Polysaccharide means the polymer of sugar. Okay, polymer of sugar. Others have steer wall made of the pure protein. Okay, pure protein. Okay, pure protein. So all the archaea lack true peptidoglycan. Okay. So even some archaea doesn't. Some archaea don't have steer wall. Okay, don't have steer wall. So some other bacteria also don't have the steer wall. Okay, so those bacteria those bacteria are either mycoplasmas. Okay, mycoplasmas. Mycoplasmas. So normally they don't have steer wall. Okay. So they use steroid in the steer membrane to stabilize the steer wall, to stabilize the steer against the lysis. Okay, like so the example would be the mycoplasma pneumonia. Okay, this is called walking pneumonia. Okay, actually. So we can use indomethacin to treat this type of bacteria. Okay, so this disease burst normally in the spring. Okay, in spring or fall. Okay, actually, so it can cause fever, malaise for the human. Okay, however, it's easier to treat this disease. We can use indomethacin to treat this disease. It's easy to treat. Okay, once we use the medication, the symptoms are gone very quickly. Okay. Low air forms. Okay, air forms. Some bacteria that naturally have the steer wall but lose it during the part of their life cycle. Okay. So they can lose their steer wall due to the infections or resistant to the antibiotics. Okay. So the gram negative outer membrane. Okay. So so the gram negative cells have 3 layers. Okay. So so the outer membrane contains polysaccharide and protein. Okay, and this is called lipopolysaccharide. Lipopolysaccharide. That means this is lipid conjugated with the polymer of sugar. Okay, as so as what I mentioned. Okay, so the sugar chain actually is used to receive the signaling. Okay, signaling. So this can be so this can work as the signaling molecule or receptors. Okay. Also they can work as the endotoxin. Okay, endotoxin. Why this material can work as the endotoxin? Because they can work as the signaling molecules. Okay, so it can be so it can be recognized and accepted by our uh, uh, immune system, by our immune system. Okay, so it can also by immune system to release the interleukin-6. Okay, interleukin-6. So, for example, T cells, B cells, once they contact the lipopolysaccharide, okay, so the T cell, B cells can release interleukin-6. So the interleukin-6 can reach to our brain, okay, to generate the fever, okay. So there's a mechanism of lipopolysaccharide work as endotoxin. Porin protein. So this protein is has the hollow structure, so they work as a channel, okay, channel to allow the water or the nutrients or certain materials get into the bacteria. So now let's talk about the cytoplasmic membrane, okay. Cytoplasmic membrane is the lipid body, okay, with the protein embedded, okay. So so the cytoplasmic membrane is very important for the lives. Okay, so it protect against the loss of the water of nutrients. Okay, it holds all the materials of the cells. Okay, it can be used for the energy reaction. Okay, actually for all the chemical reactions such as nutrients process and proteins generation, production, okay, synthesis like proteins or other substance, okay, like sugars, okay. Either for the energy, for example, mitochondria, okay, so the mitochondria provides energy through the cellular respiration. Okay, so the actually cellular respiration happened on the lipid bilayers. Okay, happened on the membrane. Okay, so they can regulate the transport of nutrients. Okay, because there are the proteins can allow the nutrients get in and waste get out. Okay, selective permeable. Okay, yes. So this is lipid layer. So it prevents the water. So it prevent water getting into. Only the porine protein can allow the water get into. Okay, so this is that means selective permeable. Okay. Difference in the cell membrane. Okay, cell envelope. So outer membrane of the gram-negative bacteria contribute to the extra barrier. Okay, because so they can protect the bacteria. Okay, resistant to certain antimicrobial chemicals. Okay, because so this material is also the lipid polysaccharide. Okay, polysaccharide. So since it is made of the lipid, it's like a wax. Okay, it can prevent certain medications to kill the bacteria. It don't allow the medication to get into, so the medication cannot kill the cells. Okay. So as because when we talk about the green staining, so we can use the alcohol to dissolve this membrane. Okay, dissolve this to destroy this membrane. However, we cannot inject alcohol to the patient's veins to kill this bacteria. Okay, so we have to use medication. That means So this layer can protect the bacteria to be killed. Okay. So that means gram-negative bacteria is more difficult to be cured by the medications, by the antibiotics than the gram-positive cell, because they have another layer to protect themselves. Okay. Alcohol can dissolve this layer. Okay. However, we cannot can dissolve this layer and kill the cells. Okay. However, we cannot inject alcohol into the patient's body to kill these cells. Okay. So because if we inject tons of alcohol, so we will kill the patient. Okay. However, we can use alcohol swab to sanitize the patient's skin or wound to kill the gram-negative cells on the surface of the skin or the wound. Okay, so we can use this. So we can use alcohol in this location. Okay, so that is good. So for the gram-negative cells, so we have to use specific medication that can cross this outer membrane. Okay, so that means so that will limit our choice of medications. Okay. Now let's work on this question. The outer membrane contributes an extra barrier in gram-positive bacteria that make them impervision. So we don't have to read the other part. Okay. So if we read this part, okay, so this is wrong. Okay, because outer membrane is for the gram-negative cells. Okay, it's not for the gram-positive cells, so this is false. Okay, so this is false. So section 4, okay, identify 7 structures contribute to the cytoplasm. Okay, so the cytoplasm is majority of the cytoplasm is water. Okay, 70 to 80%. However, it's not like a liquid. It's not like a solution. Okay, it's the gel-like stuff. Okay, it's a complex mixture of the sugar, amino acids, and salts, and serve as a pore for building blocks for cell synthesis and source of energy. Okay, so this is very important. The hereditary materials exist in the chromosomes. Okay, in the chromosomes. So DNA is aggregated in the nuclear wide. Okay. Actually, since the bacteria is precarious. Okay, so it's very small, so they don't have complex structures. So they don't need a bunch of hereditary information. So that means they are so they have only one or few chromosomes. Okay, so their genetic materials are very simple. Okay, DNA is very short. Some bacteria only have circle DNA. Okay, so circle DNA. Bacteria also have plasmids. Okay, so this is no essential piece of DNA. So actually, if the bacteria doesn't have plasmid, so they can still survive. Okay, actually, the plasmid is used as is used excuse me, is used to transfer the hereditary information between the bacteria. Okay. So so they can confer the protective traits such as the drug resistance, okay, and the toxin and enzyme protection. For example, if we treat the a patient with the diseases, so if we treat the patient with the medication, okay, since the bacteria is growing in the patient's body, okay, when we treat the patient, so the medication, antibiotics will kill the bacteria, okay. However, some bacteria can still survive, okay, so they will modify their genome, okay, to generate the antibiotics resistance gene, okay, so they will make a antibiotics resistant gene, and then so they can copy this gene to plasmid, okay, so those bacteria can transfer their plasmid contains the antibiotic resistant gene to other bacteria. Once the other bacteria uh, receive this plasmid, so they can so they can code the drug resistant gene to their genome, and then those bacteria can become to the drug resistant bacteria, okay. So so next time when we when the patient get this infection again, so we still use the same antibiotics to treat the patient, so the bacteria will not be killed by the antibiotics, okay. So that means the antibiotic doesn't have effect to kill the bacteria anymore, okay. So this is example of the plasmids. Now let's talk about the ribosome. Ribosome is the site for the protein synthesis, okay. It contains the RNA, ribosome RNA, okay. R means ribosome, okay. 60% and proteins, okay, 40%. So the ribosome so ribosome have two units. Small units is 30S, largest units is 50S. Okay, so the large and small units can combine together. Actually, it's not 80S, it's 70S. Okay, for the prokaryotes, so their ribosomes are the 70S. However, the eukaryotes also have 30S and 50S. However, the combination is 80S. Okay, so that's a difference. Okay, that's a difference. So this is 50S. This is small s. Okay, this is 50S. So once combined together is 70S. Inclusion body and macro compartments. Okay. So the bacteria use the inclusion body for the food storage, okay, or pack gas. So bacteria can use inclusion to track the gas generated in the cytoplasm to let the bacteria float in the water, okay. So the bacteria can float in the water, so they don't have to so you don't have to precipitate in the bottom of the pond or the lake, okay. And they also can store the crystal of the iron oxide, okay, because the element iron have the magnetic properties, so they can use iron oxide to lend them to aligning bacteria with according to the direction of the polarity according to the magnetic polarity of the Earth. Okay. Bacterial microcompartments. So this outer shear made of proteins arranged geometric, okay, packed for the enzymes designed to work together in their chemical pathway. Okay. The cytoskeleton. So cytoskeleton actually they are the proteins. Okay, they are supportive proteins. Okay. And it's similar to the cytoskeleton in the eukaryotic cells, okay? Arranged in the helical ribbons around cells, okay? So it can support the bacteria to support the bacteria's shape, okay? So it's also found in the archaea, okay? So it may be potential target for the antibiotic development. Now let's talk about the endospores. Okay, endospores. Endospores is very specific for the bacteria. Okay, so eukaryotic doesn't have eukaryotic cells don't have this one. Okay, it's the dormant body. Okay, produced by the Bacillus clostridium. Sporosacina. Okay, Sporosacina. So actually, it's not the endospore is not the production. Okay, it's not the production. It's not. It's not the next generation produced by the mother cells. Okay. Actually, it's still the one form of the mother cells. Okay. So. That's why we can once the cells generate the spore, endospore, so we can divide these cells to vegetative cell. That means it's like it's this part, okay? Because the endospore is the inside of the cell, is this one, okay? Endospore doesn't have any metabolic activities, so it is called endospore, okay? So the other part of the cells still has a function of metabolic activity. So this part is called vegetative cell, okay? Vegetative cell. So this is endospore, okay? Sporulation. Sporulation, that means production of endospore, okay? So why bacteria make endospore? Because the environment change, okay? So the environment is very difficult for those cells to survive, such as heat, extreme heat, dry, freezing, or radiation. Okay, radiation, or chemicals such as antibiotics. When we treat the diseases with antibiotics, so those antibiotics will make the tough environment for the bacteria. Okay, so those can kill the vegetative cells. So since the environment is very tough, so the bacteria found another strategy to survive. So they get rid of all the vegetative part. Okay, so they keep the DNA information and the important stuff in the endospore. So they yeah, so they So they hybridize. Okay, hybridize. Okay. So, for example, in this diagram, okay, in this diagram, so this is the normal cells, okay, normal cells. Once the environment change, okay, so the bacteria decide to make endospore, so they have to duplicate the important stuff. Okay, they duplicate the chromosome. Okay, so that means make 2 copies. Okay, so once the duplication is done, so the bacteria has 2 copies of chromosome. So the bacteria will use 1 copy for the endospore, leave another copy for the vegetative cells. Okay. Also, they generate a, a plate. Okay, they generate a plate to so they generate a plate from the cell membrane or cell wall to separate the cells to 2 parts. Okay, to 2 parts. However, so 2 parts. So one part is called spore. Okay, spore. Another is called sporigium. Okay. However, so this plate is not used to separate the cells to 2 cells. Okay, it's not like the sexual protection. Okay. So it's not easy to separate the cells to 2 parts. Actually, so the plates will fuse together, okay, and fuse together to let the phosphorine to be uncovered in the cytoplasm of the vegetative cell, okay. To form the early endospore. Okay, in early endospore. So the endospore contains the chromosome. Okay, that's very important for the bacteria to survive. Okay, since the endospore doesn't need the metabolic function, so they can get rid of all the other stuff such as the ribosome and all the cytoplasmic parts. Okay, and then so the early endospore can continuously develop to form the cortex. Okay, cortex. This is ultra cold layers, very thick, outer layers to prevent the tough environment to destroy this endospore. Okay. Once the outer coat becomes thicker and thicker, so the endospore is matured. Okay, endospore is matured, and then so the bacteria will generate some enzyme to degrade the vegetative cell. Okay, to degrade the vegetative cell, to lose all the vegetative cell. So and then so the endospore is released from the vegetative cell. Okay. So that means the sporangium is degraded and to be get rid of. Okay. So now, so the mature endospore is very strong, so they can resist all the tough environments. So they can, even with very dry, very hot environments, so they can still survive for several years. Okay, for several years. Once this endosperm, so once the environment changes, so for example, they have once the endosperm, so endospore get into the ideal optimal environments such as humid, warm. So so they can attract more water, get into and let the chromosome so that the chromosome to generate more and more cytoplasm stuff such as ribosome to produce more and more proteins, okay, and become to the vegetative cell again, okay. So this is the life cycle of the endospore, okay. So the medical significance of the endospore, okay. Certain bacteria have endospores, so that's make those bacteria more resistant to the treatments, okay, to the antibiotics. For example, Bacillus anthracis, so it can cause anthrax. Anthrax actually is a very fatal disease. Okay, so these bacteria can be used as bioterrorism. Okay. So this bacteria can also be used as a bioweapon. Okay, bioweapon. So now the CDC in Atlanta. So the laboratory of the CDC in Atlanta also keep the stream of these bacteria. Okay, so they can be used to make a bioweapon. Clostridium tetani, okay, can cause tetanus. Okay, so tetanus is a very fatal disease. Okay, once the patient have tetanus, many patients will die. So we don't have a good treatment for these diseases. Okay, Clostridium perfringens, okay, cause the gas gangrene. Okay, gas gangrene. So once the patient has a gas gangrene, so many patients will die of the infections. So it's very difficult to treat. Because even we use the antibiotics, so the bacteria can generate the endospores, so they cannot be killed totally. Okay, so they can still survive and cause the infections. Clostridium botulinum, okay, cause the botulinum. And the Clostridium difficile, okay, difficile, that means it's difficult to treat. Difficile is the Latin word. It means difficult, okay. So it's difficult to treat, okay. Now let's work on this question. Where in bacterial wall would you find in genetic material? Okay. So I give you 5 seconds so you guys can find the correct answer. Okay. 1, 2, 3, 4, 5. So the correct answer is the nucleoid. Okay, nucleoid. Now let's work on the section 5 or section 5. Section 5 is about the features of the 3 microorganism groups. Okay. So the first is the archaea. Okay, archaea is one type of the prokaryotes. Okay, however, so archaea were generated in the long time ago. Okay, now so they survive in the extreme environment in the earth, such as the volcano, deep sea. Okay, so we cannot find the archaea in our environment, such as in our home, in our table, in our backyard. No, so we cannot find archaea in our environment. Okay. So it is considered in a separate super kingdom, okay, more closely related to domain eukaryotes than the bacteria, okay. So archaea shares the ribosome RNA sequence not found in bacteria, okay. So protein synthesis ribosome subunits are similar. So the archaea has a unique sequence of ribosome RNA, okay. So it exhibits a novel method of DNA compaction. So it contains unique membrane lipid, steel wool components, and peeling proteins. Yeah, so even archaea as the precut said, however, it's not. It's not. So it has different features compared to the bacteria. Okay, compared to our normal bacteria. Okay. So they still have their own ancient features. Okay. So the archaea live at extreme high or low temperature, so need extreme high salt acid concentration to survive. Live on sulfur or methane, because so the archaea actually lived in extreme tough environments. Okay, so those environments are the environment in the early phase of our Earth. Okay, so at that time, so the environment on the Earth was very tough. Okay, so so that means the archaea still keep their so the archaea still keep their genetic information and structure components to survive in those extreme hot environments. Okay, that's why. So we cannot find these archaeas in our normal environment, such as our backyards, our house. Okay, so they live in the volcanoes, in the deep sea, in the North Polar, okay, like that. However, some live on human body may be capable caused by home diseases. However, it's very rare. It's very rare. Okay. So comparison of three cellular domains. Okay. Bacteria, archaea, and eukarya. Chromosome. Since the eukarya is very complex microorganism, so eukarya has multiple chromosomes. Okay, so those chromosomes can store many still store many hereditary information. So this chromosome is linear. Okay. However, for the bacteria, since bacteria and archaea, since they are very small and they don't need many genetic information, so their chromosome is so they have single or few. Okay, their chromosome is their chromosomes are very short. Okay, their chromosomes are circular. Okay, it's a range. It's a ring. It's a chromosome ring. Okay. Since their chromosome is very short, so they have to let their chromosome to be circular, because circular is circular shape can make the chromosome very stable. Okay, so the eukaryote, since the chromosome of the eukaryote is are the linear. Okay, so the eukaryote have the histone. So that's the chromosome proteins conjugated with the DNA. Okay, to make the chromosome stable. So that's the difference. Okay, so the ribosomes for these two, all these three has 50 and 30s. Okay, however, these two after communication, so the ribosome is 70s. Okay, 70s. However, eukaryote is 80s. Okay, unique ribosome RNA. Okay, so all have their unique ribosome RNA. Okay, eukaryote-like protein synthesis. Actually, we can see. So the archaea is similar to eukaryote. Okay, it's similar to eukaryote. Okay, that's because archaea had to live in extreme hard, tough conditions. So they have to have more genetic information for their to get more complicated features to live in such tough environments. That means archaea have more genetic information, so they also have more complicated organelles compared to the bacteria. Okay. So peptidoglycan, so this is specific for the bacteria. Okay, cytoplasmic membrane. So this bilayer phospholipids. Okay, bilayer phospholipids. So the archaea has the hydrogen carbon with ester. Okay, they all have ester link. Okay, steroid membrane. Okay, steroid membrane. So the specific for the eukaryote nucleus. So this only for the eukaryote because this is the major feature to classify the eukaryotic cell and prekaryotic cells. Okay, fragment. Okay, so eukaryotic cell does fragment. So bacteria has some eukaryotic cells have the eukaryotic fragment, such as the sperm cells. Okay, sperm cells. However, most of sperm cells, okay, sperm cells or the fungus cells may have fungus cells flagella. However, in the animal cells, human cells, most animal cells, human cells don't have the don't have flagellum. Okay. Now let's work on this question. Which of the following do members of the domains bacteria and archaea both possess? Okay. So I'll give you 5 seconds. Okay, 5 seconds. 1, 2, 3, 4, 5. So correct answer is 70s. Okay, 70s. They all have 70s and 30s. Okay. Linear is not linear is for eukaryote. Okay, nucleus is for eukaryotes. Okay. Fatty acid with ester link. Yeah. However, the archaea has its own specific compounds. Okay. Now let's work on section 6. Section 6 is about the classification. Okay, bacteria classification. So we use the Burgi manual of systemic bacteriology for the classification of the bacteria and archaea. Okay, so this is a comprehensive view of the bacteria and archaea relatedness. Okay, so actually, so this classification is based on the ribosome RNA sequencing. Okay. So Berge manual of determination bacteriology based entirely on the phenotypic, okay, based on the phenotypic characteristics, okay, such as the shape, size, color, also the flagellum, okay. So so it is also based on the treat, okay, treat. So we can so the treat we discovered on clinic research, okay, such as shape, okay, in microscope, or the metabolic characteristics, such as the end products, what end products they made, okay. Taxonomic scheme, okay, taxonomic scheme. So once we classified the bacteria, so we can give them a name, okay. So we can divide them to different groups. We can divide the 4 major divisions of bacteria, okay. So the Gram-Silicus is gram-negative with the thin, gram-negative with the thin, okay, thin cell wall. Formicus is gram-positive, okay, gram-positive with thick, strong cell wall, okay, and Tenericus means no cell wall, soft, okay, and Mendelsicus actually they are the archaea, okay, archaea, primitive cells with unusual cell wall and nutrient habits, okay, yeah, because the archaeas are unusual, okay, that's why they can survive in the tough environments, okay. Bacterial species, okay, bacterial species is a collection of bacterial cells, all which is, share overall similar patterns, okay. So all the bacteria actually can share at least 70 to 80% of their genes, okay. They have so 70 or 80 of their genes are same, okay, are same. Subspecies subspecies and treat, okay. So the bacteria of same species, so the even in the same bacteria species, however, some bacteria may have their own specific features, okay. So we can divide those bacteria based on their specific features to the subspecies or strain, okay. Serotype, okay. Once the bacteria invades the animal humans, so they can generate the antibody, so they can cause the antibody, so they can cause antibodies generated by the animals or humans, okay. So when we detect these antibodies, because those antibodies is specific for the bacteria, okay. So we can use these antibodies to classify the bacteria. So this is called serotype. Okay, serotype. Now let's work on this question. You identify a bacterium as gram-negative. Use gram stain method. In Burge menu of determinative bacteriology. This bacterium belongs to what? So I give you 5 seconds, so you can give me the correct answer. 1, 2, 3, 4, 5. Okay, the correct answer is A. Okay, Gram-silicus. Gram-silicus means gram-negative. Okay. Formicus means gram-positive. Okay. So Tenoricus cannot be stained by the gram dye. Okay. So that means no. So it's not like acid fast. Okay. Acid fast bacteria like Mycobacterium tuberculosis. Okay. So the Mendelsicus actually, so that's for the archaea. Okay, archaea. So we have to. So this is also not for the gram staining. Okay. Okay. So this is the appendix of these chapters. So you guys can use this appendix for your reviewing of reviewing all the contents of the chapters. Okay, so you guys please feel free to review these chapters. I hope my lecturing will make you master the knowledge of this chapter easily. Okay, so this is the end. Thank you.